The overall goal of this procedure is to produce a mouse model of oral cancer that allows the visualization and quantification of tumor invasion in the tongue. This is accomplished by first generating oral squamous carcinoma cells for 2-photon imaging by lentiviral infection of LifeAct M. cherry and stable clonal selection. Next, orthotopic xenograft tumors are produced in nude mice by injection of LifeAct M. cherry labeled cells into the tongues of anesthetized mice. Then, tumor-containing tongues are imaged by 2-photon microscopy. Ultimately, results can be obtained that show quantified local oral tumor invasion within the tongue muscle through 2-photon imaging of fresh tongue tissue and subsequent computer-aided 3-dimensional reconstruction of primary tumors and regionally invaded cellular groups.
The main advantage of this technique over existing methods like immunohistochemistry or bioluminescent imaging is that tumor cell invasion can be quantitatively measured in 3 dimensions. The implications of this technique extend towards therapeutic intervention in oral cancer because it provides a model system for the analysis of proteins involved in tumor invasion as well as the assessment of preclinical compounds on anti-invasive therapeutic strategies.
Generally, individuals new to this technique will struggle, as injection of the tumor cells into the tongue and preparation of the tongue for 2-photon microscopy is technically demanding, requiring skill and practice. Visualization of this method is critical, as the tumor injection and 2-photon steps are difficult to learn and require specialized skill.
To begin, culture human head and neck tumor cell lines in complete medium consisting of DMEM supplemented with 10% FBS, 1% penicillin streptomycin, and 1% non-essential amino acids. In order to transfer the LifeAct M. cherry coding sequence into the PLL 7.0 lentiviral vector, site-directed mutagenesis is first required to introduce 3 silent mutations into the SBF1 recognition site in the parent M. cherry cDNA. Next, PCR amplify the modified LifeAct M. cherry sequence with flanking EcoR1 SBF1 sites and subclone it into the PLL 7.0 to generate a PLL 7.0 LifeAct M. cherry construct. Following a lentiviral expression system, culture the packaging cell line 293T17 to 40% confluence in complete medium. Using CalPhos, transfect the cells with the PLL 7.0 LifeAct M. cherry, PSPax2, and PVSVG vectors in a 3:2:1 ratio, respectively. After 24 hours, replace the medium with fresh, complete medium. Collect and replenish the medium every 12 hours for 72 hours, and store the collected medium at 4 degrees Celsius. To produce head and neck cell lines with stable LifeAct M. cherry expression, spin the collected medium at 2000 RPM for 10 minutes at 4 degrees Celsius. Add 1 milliliter of the clarified medium containing the virus to the OSC19 or UMSCC1 cells for 12 hours. Rinse the cells, then add an additional 1 milliliter of virus for another 12-hour period. Culture the cells in medium containing 200 milligrams per milliliter of puromycin for 2 weeks to select resistant colonies. Visually screen surviving colonies for LifeAct M. cherry expression by fluorescence microscopy. Trypsinize individual positive colonies using sterile 3mm cloning discs. Maintain positive cells in medium containing 200 milligrams per milliliter of puromycin until frozen back or used for orthotopic injection. To generate an orthotopic tumor xenograft, trypsinize LifeAct M. cherry expressing tumor cells, then centrifuge the culture and resuspend about 2.5 times 10 to the 4th cells in 50 microliters of complete medium. Load the tumor cells into a 1 milliliter syringe attached to a 27 gauge half-inch needle. Next, anesthetize 8-week-old female athymic FOX1 nude mice with a combination of 80 mg per kilogram of ketamine and 10 mg per kilogram of xylazine. Once the animals stop moving, apply eye lubrication ointment and check for responsiveness by pressing a fingernail into the foot pad. Maintain the mice on a heating pad between 37 and 40 degrees Celsius. Using sterile forceps, gently grasp the tip of the tongue and pull it out of the oral cavity. Slowly inject the cells into one side of each tongue to create a bulbous mass in the tongue center. Inject the mice with 2.1 milligrams per kilogram of yohimbine and return them to the heating pad to monitor them during recovery from anesthesia. Place mice in sterile cages containing a soft transgenic dough diet. Weigh the mice every 2 to 3 days and monitor them visually for tumor onset. To prepare mouse tongues for ex vivo imaging, euthanize mice harboring tumors at different time points using carbon dioxide inhalation. Extract the tongues and rinse them with 1x PBS. Then, using monofilament sewing thread from a local hobby shop and a size 8 sewing needle, attach the tongue to one side of a conventional paraffin tissue embedding cassette. Once the tongue is immobilized, submerge the entire cassette assembly in 1x PBS. Immediately process the tongues using 2-photon microscopy. To image the tongues using 2-photon microscopy, submerge the tongue cassettes in a 60mm dish containing 1x PBS. Secure the dish in a custom-designed holder on a retractable cantilever arm positioned under the objective of the 2-photon microscope. Place a 40x 0.8 numerical aperture water-dipping objective lens directly on or over a visible tumor lesion. Image the tongue by 2-photon microscopy with a titanium sapphire laser with an intensity of 60 milliwatts and an input wavelength of 755 nanometers to optimize the M-Cherry signal. Collect serial 1 micrometer laser scanning images at 1 micrometer incremental depths over a total tissue depth between 15 and 100 micrometers. With this configuration, tumors up to 1 millimeter in tissue depth can be analyzed. Use Scan Image to capture images. Scan Image generates a 2-channel output of raster scan patterns to control XY galvanometric scan mirrors, and at the same time captures a maximum 4-channel signal input simultaneously from photomultiplier tubes through a data acquisition board. Scan Image collects Z-stack images by controlling the Z-axis of the objective, and collects time-lapse images in a single or cyclic mode. Save the images in a single TIFF file with 16-bit depth. Using the Amira software, render tumor image stacks into a single 3-dimensional image by opening the TIFF file containing the set of Z-stack images. Use the Voltex function to generate a 3-dimensional rendering. A large primary tumor image with several smaller, dissociated invasive groups, or IGs, will likely be present in the rendered image. To measure the tumor volume, define the thresholded primary tumor area in each slice of the image stack. Correct for and eliminate background fluorescence. Repeat the thresholding procedure for each invasive group. Once the primary tumor and all the IGs are selected, determine the volume measurement as well as the X, Y, and Z tumor centroid coordinates for the primary tumor in order to calculate distances of IGs from the central tumor point. Calculate the tumor invasive index, or TI, using the formula TI equals NT times VT times DT, where NT equals the total number of IGs in the image, VT equals the total volume of all the IGs, and DT equals the total distance traveled by all invasive groups from the center of the primary tumor. 3-dimensional renderings with greater topographical detail can be generated by importing the original 16-bit monochrome TIFF files into Nikon NIS elements. Within the software, create an ND file from an image stack. Then manually calibrate the document to specify the pixel size. For higher quality renderings, add additional slices in the Z-plane. This figure shows an example of the raw data acquired from a 2-photon image. NIS Elements or Amira software can be used to reconstruct the raw data of tongue tumors, which is shown in this figure. This panel shows an example of the 3-dimensional rendering using the Voltex function in the Amira software. Here is an example of thresholding of an individual 2-photon section from the image stack to identify invasive groups from the primary tumor, as well as the primary tumor center, or centroid. After each thresholded section is analyzed, Amira quantifies the volume and distance traveled by each invasive group from the centroid. These data can be graphically demonstrated and used to obtain the numeric value for the complete level of tumor cell invasion attributable to the primary site. Shown here are representative images of tumors as visualized by conventional pathological immunohistochemistry labeling compared to a 3-dimensional image of a similar tumor using the described protocol.
Once mastered, from the point of tongue extraction to the final 3D rendering, this technique can be done in about 2 hours, if properly performed. While attempting this procedure, take care not to puncture the tongue through with the needle, thereby losing tumor cells and impeding the tumor take.
Following this procedure, testing of therapeutic compounds or cells with manipulated protein levels can be conducted in order to determine their efficacy on oral cancer invasion in an in vivo setting.
Don't forget, working with human tumor cells modified with lentivirus is hazardous. Precautions, such as proper PPE and working in a BSL-2 certified laminar flow hood, should always be taken while working with injected mice.